So let's switch in English now. For the first Plastic in the Ocean talk, so it's my pleasure to introduce Valentin Dettling, a PhD student that I co-supervise with Sarah Samadi, and he is working on microplastics, and microplastics that can be detected in some sea cucumbers. And there are multiple things that could be done on that, so I let you show us what is about these sea cucumbers.
So thank you, Jean-Baptiste, for the introduction. And thank you, everyone, for being here today. So I'll be speaking in English. I hope for non-English speakers it would be okay to follow. And at the end, I will gladly accept questions as well in French. So as mentioned by Jean-Baptiste, my name is Valentin Dettling. I'm just finished my first year of PhD here at the French National Museum of Natural History. And the subject of my presentation today is what can specimens from natural history collections tell us about the temporal evolution of plastic pollution. So, as you may be aware, we are now in a world that is contaminated by plastics. And this can be due to the fact that plastic production has increased a lot in the recent years and has even become exponential since the 70s. And the result of that is that there is actually a leakage of this plastic production into the environment. So there is a leakage of plastic waste. And it is estimated that, for example, for the year 2010 alone, 4 to 12 million tons of plastics have reached the oceans. Everywhere we look for them, we find them. They are found in very remote areas, such as Arctic polar circles, or even in the human body. In 2018, Peng and colleagues even showed that microplastics could be detected in the Mariana Trench, which is the deepest trench in the ocean, that goes up to the depth of 11 kilometers, So which is a place in which humans have never been even before, but still we managed to contaminate it. So once plastic enter the environment, they don't remain as they are. They undergo a weathering process. And if you take a look on this plastic bag, well, I can't look and talk in the. So if you take a look at this, okay, I won't do it. At the plastic bag, which can be a waste that you find usually in the environment. If you look closer, plastics are usually polymers. So it's an assembly of little bricks that are called monomers. And those bricks are also, during the synthesis, there is additives that are added in the plastic items to make them more rigid or less rigid, for example. And due to time and environmental factors, such as the salt of the sea or the sunlight, the plastics degrade themselves into smaller particles that are called microplastics, if their size is comprised between 5 millimeters and 1 micrometer, and even smaller ones that are below 1 micrometer, that are the nanoplastics. And among other things, microplastics, they have and they will end up in the deep ocean sediments, and this will increase even more in the coming centuries, even if we stop the plastic production today. And so they reach the deep ocean sediments, mainly because either of their density, they are heavier than seawater, or due to biofouling, that is a process that consists in the aggregation of microbes or algae on the particles. And so actually, when we talk about plastic pollution, we're not talking about a single pollutant, but actually we are facing a family of pollutants. If I may say, it's a myriad of pollutants. And this is due to the fact that there are many types of plastic that exist. You may be already aware of those logos here, such as PET for water bottles, for example, PVC or polypropylene, and also many types of additives that can be added, such as bisphenols or phthalates. And so as we are facing a contaminant that is multiple, we are also facing consequences for marine organisms that are as well multiples. And so these consequences can be dependent either on the size, on the shape, on the polymer, or even the color of the polymer. And they can be as various as choking of the animal, malnutrition, inflammation, and so on, and even consequences that we don't know yet. So let's move on now to why I'm going to talk today about natural history collections. So here you are at the French Natural History Museum, and we are lucky in such museums to have access to what we call natural history collections. So these collections are actually archives of samples that have been collected in the past, so for the past centuries, and these collections have been constructed thanks to expedition in the field, for example. Here at the MNHM, we have more than 60 million specimens, so it's quite a lot. And under the Jardin des Plantes, just below, not here, your feet here, but if you stay close to the Grande Galerie de l'Évolution, there is a library of zoological specimens that you can see on this picture. And in this library, you can find specimens of many types of animals that are stored either in ethanol, formal, or dried. You can even, so I just show a close-up on sea cucumber. So this is one from 1944 that is quite well preserved. So these collections, they have been originally created with the goal of describing and studying the biodiversity. This by taxonomical methods. However, today, researchers start to find new uses of these collections, and they are thought to be witnesses of past changes. And by past changes, maybe we can include anthropogenic factors. So today we're here to talk about plastic pollution. So the idea, our idea, and the idea of other researchers would be to use these collections with the hypothesis that the plastics that are found within these organisms maybe reflect the increase of plastic pollution. So I'm going to try to show you 3 examples of studies that already used such collections to perform such analysis. So for example, in the Atlantic, Curtin-Jones and collaborators use collections of sea star and ophiuroids. And they used the samples that were collected between 1976 and 2015. They show, actually, that the ingestion rate, so the microplastic they found in the sea stars, were consistent across time, and that the type of polymer were also not variable. Nonetheless, another study made by Hu and collaborators that this time used fish collections showed that between 1900 and today, there was an increase of plastics in the fish. So for 1900, it could be logical because plastic was not very well used. And they show a very interesting figure in which they compare the quantity of plastic they found in the fish, and it kind of follows the accumulation of plastic in marine sediment as well as the rate of increase of plastic production. And finally, another study by Halbach and collaborators used mussels from the Baltic Sea and the North Sea between 1986 and today, and they show either an increase or a stagnation of the quantity found in the collections. So from all that, you can be confused because they use various organisms and they lead to various conclusions about the evolution of microplastic quantity. There can be many explanations for that. So first, they use different organisms, so maybe they don't all reflect what's happening in the environment. They also use 3 different methods to quantify microplastic, and they also don't look all the studies doesn't look at, they don't look at the same size fraction. For one study, for example, they only study microplastics that have a size of above 50 microns, and for another one, it's above 1 micron. So this could also explain the difference. Finally, those studies they don't always take care about the sample contamination, meaning that for these collections, we don't have blanks and we don't have controls to be sure that what we are seeing is actually a real signal and not a contamination due, for example, to the fact that researchers at the time of collect used plastic gloves and contaminated their sample. So through our project that I will present to you today, we'll try to answer 3 questions. So first, can we detect microplastic using our collection here, and how? I tried to show you 2 evidence and 2 concerns that we have about the contamination of our samples, and also a first look into the location of those microplastics in the animals. Here at the museum, there have been many expeditions that have been led, and one of the programs is called Tropical Deep Sea Benthos expeditions that have been led since 1976 and that are still going on. And those expeditions have been principally done in New Caledonia because it's a region that is very rich for its biodiversity. So here we have the chance to have access to collections of specimens that have been sampled in the deep sea over 40 years regularly at the same place. So you may already see what I'm trying to do. What we are trying to do here is to use these specimens, a bit like the other studies that I showed you, to try to qualify and quantify microplastic in a retrospective manner. So I'm going to try to guide you through the journey in which we have been embarked since the beginning of the project. And so the question is, how do we proceed from here? We want to detect microplastics using collections, but it seems rather easy, but it's not so easy actually. So there is a first step of specimen selection and another one of microplastic quantification. So we had to choose a taxon, which is an organism. Then we had to look for the samples to see whether we knew which species they belong to before doing a choice. And after that, once you've performed all of those steps, you can select a method and adapt the protocol to be able to reach the goal of quantifying, I can't do both really, sorry, quantifying the microplastics and analyzing the type of plastic you have across time, sorry. So first step, which taxon? So the organism that you choose for doing such a study really depends on your research interest and questions. In our group, here at the museum, we're interested into benthic ecosystem ecology. So this is the ecology of organisms that live close to the seafloor. And as a result, as Jean-Baptiste mentioned in the introduction, we got very interested by a not so beautiful organism, that is the sea cucumber or holothurian. So those organisms, maybe you already saw some of them when you go on the beach, actually close to the beach. So they are present at every latitude and depth of the world. And they are so much present that if you take the global biomass of organisms that live on the seafloor, at some point they represent the majority of the biomass from the seafloor. And they are kind of they are a very strange species because to eat, they extract organic matter from sediments. So basically what they do is that they eat, sort of, the sediments. It goes through their digestive tract, and from that they extract the organic matter. And this process is called the bioturbation because they mix the sediments, and they do this at a very, very high pace because for one individual, it's between 9 and 80 kilos per individual per year. And we have evidence in the literature that microplastic have been found in their internal organs, either in the field, from field sampling, or with exposure experiments in laboratory. So our hypothesis is that sea cucumbers could be a relevant organism to study benthic plastic pollution. Now that we had chosen our organism, we needed to look for the samples here at the zoological library. However, we don't have the chance today to have at the museum a specialist of these organisms, and as a result, there was not much information available on our database So to make it short, we didn't know where were the specimens for most of them, and we didn't know which species they belonged to. So we had the first job of locating and grouping all of the barrels and the jars, and some of them are even not here. We located them at the lab of collaborators in Russia, which complicates things a bit. And so for this, after we have located all our specimens, we established a workflow that was very tidy work, and we thank Elias, that was our intern for that. So he had to take each specimen, individualize it, give it a number, photograph it, and take tissue sample, while making a database of all of this information. Here on the right, you can see a photograph of a specimen that is quite in good shape, actually, from the 1989 campaign. In the end, he did that for 1,200 specimens, so which was a long work to do. And so if I just show you where they come from, so as I mentioned that tropical deep sea benthos expeditions were mainly led in New Caledonia, so we targeted this region, and we have specimens that are coming all from along the coast of New Caledonia, but also from more remote areas. Yeah, you can see it, for example, on the right-hand side. So we have a good geographical coverage and also a good temporal coverage. So here are the campaign names. So the specimens come from very different campaigns. And if we take a look across time, you can see that we have a coverage that goes from 1984 to today. We mainly have samples from today, but still we have a few hundreds that come from the 80s and the 90s. Sorry. Then the next step was to know whether the species were identified. And what we mean by that is to know whether we know which species they belong to. So we took a tissue sample of every organism. And thanks to integrative taxonomy, which is the combination of multiple methods to identify species, we either will perform barcoding, which is a molecular technique using DNA, to identify the species thanks to a database, or more classic taxonomy, that will be by looking at speckles that are tiny calcification present in the tegument, so that is the skin of the sea cucumber. And this will be done also with the Chershov Institute in Russia. I have first to talk about results a little bit. We started to do the barcoding, so we are only at this step of the project. And so this is the example of two different species of sea cucumber that are, I don't know which one you prefer actually, that are very, very different, and for which we didn't know the species they belong to. But thanks to the barcoding, so we took a sample, we extracted the DNA, and we compared it with the barcode of life database. We were able to say that the specimen from the left is from this species, and the other one from the other. We are quite confident because we had an homology of the sequence between our sample and the database that was above 98%. So if I sum up for this part, we performed, thanks to the work of many people actually, to the collection team and Elias, as well as Claire, that was a postdoc with us, the inventory of the specimens that we have at our disposal, and we started to perform the identification. This way, we'll have a clean database in which we'll have all our specimens with the year they were sampled and species identification. But now, which specimen do we choose? So I'm afraid I won't be too specific today for this part, because we have our 1,000 specimens, we'll identify them, and then we'll have to select some of them because we want to quantify the microplastic in each of those specimens. And after we've done this work, thanks to the help of Claudia also as well, who is an engineer in the team, we'll select around 60 specimens that are representative, and so we'll try to find 2 species that are found multiple times in our chronology, and that come from different regions. So for example, one from the coast and one from a more remote area. But maybe we won't have the luxury of having two species that we found exactly at the same species exactly across time. So if it's not the case, we'll use clusters of similar species. But this will really depend on what we have in the end. So now that I've talked about the specimen selection, we're not waiting actually to have chosen our specimen to know which method we'll use to quantify microplastics. And I'm going to show you some preliminary results. But we had to define a method. And for that, we established a collaboration with Alexandra Terhal, who is a researcher at the IMRCP lab of Toulouse, and who actually had the same idea of project. So we decided to join our forces to do only one single project. So we have the specimens, and they have the method that they published in 2 papers in 2022 and 2023. And so we started from a working protocol that was for organisms. So you have over there, so here your sea cucumber. And so you have to freeze dry it. In French, it's lyophilisation. And then you break it, and so you have a powder. This powder is digested using KOH and then filtrated using a filter. This filter retains everything that is above 1.6 micron, which is quite a low cutoff. And in the end, you obtain your microplastics and other things as well on a filter. That way, thanks to the method they developed, we can detect and quantify microplastic by mass spectrometry. So if we zoom on the method, so you have your biological sample that goes through multiple machines, sort of. That is pyrolysis, gas chromatography, and a mass spectrometer. And all of this process enables to detect specific markers for each of the polymers that are listed here that represent actually 6 types of plastic that are mostly found in the environment. So these markers, if we detect them, we are very, very sure, thanks to all of this process I won't go through, that you are in presence of this type of plastic. And thanks to calibration curve, so meaning that we have standards of which we know the concentration, we are able to quantify the quantity of microplastics of each type in one run for each sample that we have. It's a very sensitive method because the limit of quantification is of 25 nanogram, which is quite low. They already published results in 2022 using organisms from the seafloor, and they were able to detect 4 types of plastics in those organisms. But it's not funny if it's already working when we started the collaboration, so we had to adapt the protocol because sea cucumbers, as I mentioned, they eat sediments. So when you have them in the collection, you have sediments in their guts. So it's kind of annoying because it's usually not the same methods that are used to extract microplastics from sediments or from organisms. But we're still trying to adapt the protocol so that it would work. And I won't go through every step, but one thing that we decided to adapt first was not to analyze the full specimen, but to dissect it and only take the gastrointestinal tract, so the intestine. And the main reason is that transit and storage of our specimens was carried out so they often end up in glass jars, but in the process, they are transported in barrels, Ziplocs, or metal drums that all contain polyethylene that could actually contaminate our sample. So for the moment, we are doing a workaround of dissecting the specimen to only take the inner parts that are not in direct contact with potentially contaminated ethanol. We are also we are doing the quantification of the quantity of plastic that we find in this ethanol to be sure it's not too high. And so before using our precious collection specimen, we started to assess whether the protocol was working using samples that we took from Marseille and Concarneau, so with the help of the Marine Station of Concarneau and Didier Aurel from Marseille. So they had the kindness of going and take specimens that were close to the Bay of Marseille, the Bay of Concarneau, And we did the protocol I just told you about and detected and quantified microplastics. These are preliminary results, but so what you can see here, so the colors represent different individuals. The first two ones are from Marseille and the other two are from Concarneau. And in the y-axis, you can see the polymer concentration that is in a logarithmic scale. I won't go through the details of this, but what you can see is that we can detect almost every polymer except PMMA in those organisms. So we were already happy with that because we can see plastics in our sea cucumbers. Also, the concentration that we found are consistent with the literature. Nonetheless, you can see that for polyethylene, you have a very, very high concentration that is on average 30 to 50 to 100 times higher than the other polymers. Maybe this is due to the fact that we use polyethylene everywhere in the zoological library. So maybe we won't be analyzing this fraction, but we are not sure yet because polyethylene is frequently found in high levels in those organisms. I'm going to finish this presentation by showing you two very preliminary results as well about our try to locate where are the microplastics in sea cucumbers. So we are working with collections, so we try to use non-invasive techniques. There is a part of the project also where with the Marine Station of Concarneau, we want to do experimentations where we feed the sea cucumbers microplastic and to see where the microplastic go in their organism. So we established a first collaboration with the platform Asterix from the museum with the help of Marta Bellato. And we managed, using CT scan, that is like in French, scanner, so using X-ray, first of all, to see the internal contents, so the organ of the sea cucumbers. So here on the picture that will then move, you can see mainly the intestines. So we managed to see it, which was the first victory, actually. And then we artificially injected plastics that were radio-opaque inside the sea cucumber. And as you can see on this video, that is, so we go all along the sea cucumber. The white dot that you see are the spheres that we injected. So they are visible through the tissue. And what you must get from that is that we didn't open the specimens to see all of this. So this is interesting. We won't be able probably to use it for collection specimen because sadly the plastic spheres are not radio-opaque, but it still remains an interesting result. We also established another collaboration with Ipanema platform with Mathieu Toury. It was an experiment in which we took out the intestinal tract of the sea cucumber and injected this time fluorescent plastics inside the lumen of the intestine. So here you can see that you have some of them that are outside because we injected a lot of them, but actually the ones that you see inside the tissue, they are really inside the tissue. So actually the fluorescence goes outside. So it's a semi-invasive technique because we didn't have to cut in small pieces the gut, but still we had to take it out, so it's a bit invasive still. And this as well remains to be investigated further. So I'm a bit late, but I'm going to conclude here. I hope today that I kind of convinced you that using natural history collection could be interesting to study microplastic pollution in a retrospective manner, and that it seems feasible. Nonetheless, we are very, very aware that we have a lot of contamination that can be seen in our samples, and we try at the maximum to not take it into account. A perspective would be to include more taxa, so more organisms in our study, to have a global understanding of the spread of microplastic contamination levels as well as their consequences. Thank you so much for listening to me today, and thank you to all of these people, because I was talking about this project today, but I'm not the only one working on it, so I would like to warmly thank everybody that participated. Thank you.
Thank you, Valentin. On peut prendre des questions en français aussi.
Ouais.
Okay, Valentin, thank you for the presentation. I'm Sylvie Dufault, chargée de mission mère au Muséum national d'histoire naturelle. Concerning your model holoturies, do you know what is the lifetime of the holotury? To have an idea of how long they can accumulate during their life, the plastic you will measure at the time.
Yes, so we are talking about a lifespan that goes from around, in average, it could be like 7 years, but it can be more or less depending on the species. Because sea cucumbers is a bit like if we talk about birds. It's a class of organisms, so it's very large, even though they all look the same, almost. So yeah, but the lifespan is in years.
And do you have a way to determine the age of the samples you are investigating?
So it's actually at Concarneau they are very interested into being able to determine the age. They just managed to have clues, but actually no, we are not able to know, and that is sad because actually it would be very nice to know the age of the specimen. So no, we really can't decipher between a small individual or an old individual.
Can I? One question? My question is also for the samples of the collection. Do they collect at the same time seawater beside the sample to check the microplastic in the surrounding environment of the sample?
You mean in the collections?
Yeah.
So sadly, we hoped that during the expeditions they would collect sediments for geologists, for example, or water. Actually, it hasn't been done. So we won't be able with our study to say that what the quantity we find is reflect of what we have in the environment. Nonetheless, we performed one expedition in New Caledonia last year and another one next year. And the goal is to also take water and sediment to at least have one time point to see whether the concentration is proportional.
Okay, that's important.
Yes, completely.
Can I have one last one? Do you plan to work also with oceanographer to have an image of the currents and to determine if the plastic may come from New Caledonia itself or from major ocean currents?
So actually, it's not. I mean, I believe it would be interesting. We didn't yet have established such a collaboration, and we tried to look into the literature about currents close to New Caledonia, and there's actually one study that evaluates the particles that could flow in this area. So maybe it could be interesting to establish a collaboration with the people working there. But to be honest, we didn't yet do it.
And about the water samples, it's not a trivial question because we had to choose before the last expedition if the samples had to be done, at the surface or at the benthic zone or in the middle. So it's something that we need to find out.
Merci beaucoup pour ta présentation, c'était vraiment très très bien. Je pense qu'un paramètre qui va être très important dans ton interprétation, ça va être la quantité de sédiments dans le concombre. Parce qu'en fait, ce qui va se passer, c'est que tu as deux signaux qui se mélangent. Tu as le signal du concombre qui broute, donc c'est l'accumulation, versus la quantité de sédiments que le concombre a. Et donc, dans des cas où si tu n'as pas les mêmes ratios sédiments, chair de concombre, tu vas enregistrer un coup une pollution de fond versus une pollution intégrée. Et donc, je ne sais pas si ça varie énormément, mais je pense que c'est quelque chose que tu dois intégrer.
Oui,
c'est que tu avais imaginé ça.
C'est ce que on essaie de trouver un moyen justement de pouvoir séparer les deux parce que c'est pas évident de pouvoir enlever les sédiments de l'intestin. Mais effectivement, et puis même avec notre collaboration à Toulouse, nous on avait l'idée de regarder la bioaccumulation à la base et eux avaient l'idée d'utiliser le concombre de mer comme un petit sac qui contient des sédiments. Donc déjà, c'était pas la même question qui était derrière.
Je pense que prendre ce concombre pour dire que c'est du sédiment, c'est peut-être plus la vision que de l'accumulation. Parce que tu connais pas. En fait, on ne connaît pas encore ce qu'on appelle la dépuration. C'est-à-dire qu'il se peut que le concombre, ça soit juste une phase transitoire, tu vois,
Oui.
c'est-à-dire ça broute, ça stocke X, et puis ça,
Oui, tout à fait.
dépure. Et donc, ça se trouve, ça
fait que passer, ça se trouve.
Que ce que tu as le sédiment à
Oui,
l'instant T.
bah, c'est pour ça que du coup, les expérimentations qu'on veut faire à Concarneau en nourrissant les concombres de mer avec du plastique, on veut essayer de faire des expérimentations qui durent si on peut, sur le long terme. Je ne sais pas si on aura le temps durant ma thèse, mais en tout cas de pouvoir avoir ces indices-là qu'on n'a pas trop dans la littérature sur les concombres de mer, en tout cas.
Hello, Anni Herranen, postdoc from the Museum. I was wondering, do we know how well the plastics are preserved in the ethanol and in the samples?
Sorry, I'm not sure I understood. You mean the sea cucumber or the plastics?
The plastics.
If they are well preserved in ethanol?
I understood that your samples are conserved in ethanol.
Yes, yes.
So ethanol, does it dissolve plastics? Or?
No. There has been studies in the literature showing that actually ethanol doesn't degrade the plastics. Yeah. So yeah, we made sure of that as well.
Okay, thanks.
No question? No more questions? You'll have the time at the coffee break too. So thank you, Valentin.